Good morning children, I am Sardaisis. Now I am going to teach you central dogma. Before getting to the details of central dogma, we have to see some basic informations in regards to DNA. Let's see. DNA, as you know, it is made of two polynucleotide chain, and the DNA is made of two polynucleotide chains, and the backbone of these two chains are sugar phosphate. So sugar phosphate make the backbone of this DNA, and the nitrogen bases project inside. Nitrogen bases project inside. And which are the nitrogen bases? Two nitrogen bases, purines and pyrimidines. Purines and pyrimidines are the nitrogen bases project inside. The purines, which are the purines? Adenine and guanine. Adenine and guanine are the purines. And which are the pyrimidines? Thymine and cytosine. These are the nitrogen bases. And this purines always base pair with the pyrimidines. The adenine base pair with the thymine and guanine base pair with the cytosine. In turn, we can say the thymine always base pair with the adenine and the cytosine always base pair with the guanine. Okay, so this is in case of DNA. Adenine, guanine, thymine and cytosine are seen as nitrogen bases that project inside. But in case of RNA, instead of thymine, uracil is there. So in RNA, thymine is replaced by uracil. Okay, so that is the difference between DNA and RNA. In detail, I will teach you later. Next, we can see the central dogma. So, the definition of central dogma is the proteins which are synthesized by DNA through RNA. This process is called central dogma. How the DNA can synthesize an RNA strand. So, before seeing that, we have to see how a DNA can synthesize a DNA strand. Next, we can see how it forms RNA strand. Okay. First, we can see, suppose there is a double-stranded DNA. So, this is a single strand with the polarity 5 prime, 3 prime, and another strand which has the polarity 3 prime, 5 prime. So they are anti-parallel. Different polarity they have. And nitrogen bases we have seen inside. Nitrogen bases. Suppose this is adenine. This must be thymine. This is cytosine, means this must be guanine. Again thymine means this must be adenine. Like that, there is a base pairing inside. Okay. And these two strands, when it gets unwound, we can see the first strand 5 dash or 5 prime, 3 prime. So another strand 3 prime, 5 prime. This is the unwinding stage of this double-stranded DNA. So suppose here A, C, G, G, C, G. This is the nitrogen bases, and that must have the complementary base pairing. So this must be T, here G, here C, A, here C, here G, and here A. Okay, so this is the base pairing. And here, this strand, these two strands act as template strand. Template. What is the meaning of template strand? Template strand means a a single strand can make a copy of itself. It can synthesize a new strand. Suppose this single strand can synthesize a new strand, and that new strand must have the complementary base pairing. Let's see how it forms. First we can see this first strand as a template one. So these two strands act as template strand. Template strand means a strand which can synthesize the copy itself, and that strand is called a template strand. Here in the DNA strand, the two strands can act as template strand. That means the two strands can synthesize their own copies, or the two strands are capable to form new strands, and that must be complementary to each other. Let's see the first strand here. So this strand is the parallel strand. We can grab this. So this is the parallel strand. So this is the parallel strand. From this parallel strand we can take this single strand here. This single strand with the polarity 5 prime, 3 prime. Okay. And what are the nitrogen bases? A, C, T, G, C, T. Okay. And this strand can act as a template strand by forming a copy of itself. It must produce another strand. It can produce a copy of itself, and that must be complementary to each other. So this consists of thymine, and this must have guanine, and this adenine, this cytosine, this guanine, this adenine. So this is the new strand, new complementary strand with the anti-parallel polarity 5 prime. Okay. So this single strand we are taking here, and this single strand acts as a template by forming a new strand, and this must be complementary to the template strand. And we can consider, or we can see this strand. Okay, this strand, the next strand. So this is the next strand. Here we can write over here. So what is the polarity? 3 prime, 5 prime. And what are the nitrogen bases? Thymine, guanine, adenine, cytosine, guanine, and adenine again. So this strand we are taking as a template over here. Next we can see what are the possibility of having nitrogen bases in the newly formed strand. So this strand acts as a template strand by forming a copy of itself. So it is producing, here we can write, it is producing a new strand, or it is synthesizing a new strand with the nitrogen base T, so must be adenine. Here must be cytosine, because thymine always base pair with the adenine. Here thymine, here guanine, here cytosine, here thymine. So I taught you the adenine always base pair with the thymine with 2 hydrogen bond. Here 3 hydrogen bond. We can draw the bonding over here. Anyway, so these are the newly formed strand from the parallel strand. So these two strands are identical to the parallel strand. Am I right? Let's see. These two strands are identical to the parallel strand. And this process of formation of a new copy or new strand from the existing strand is called DNA copying or DNA replication or DNA replication. Okay, so the DNA, the two strands of DNA can act as a template strand by forming or by synthesizing a new strand, and which are complementary to the template strand. And this is called DNA replication or DNA copy. In case of DNA replication, both the strands are involved in the synthesis of new strand. Okay. Next we can see the central dogma in detail. Here actually the central dogma means from the DNA, the genetic informations are passed into RNA. Okay, and from the RNA, the genetic informations are passed to form protein. Okay, so the DNA itself replicates. I taught you it's a replication. Replication means it can form their own copy. Okay, so this DNA, there is a flow of genetic information from the DNA to the RNA. Here it is mRNA, messenger RNA. The type of RNA I will teach you later. So here the flow of genetic information from the DNA to the mRNA, and that process is called transcription. So what is transcription? The process of flow of genetic information from the DNA to the mRNA is called transcription. And there is a flow of genetic information from the mRNA to the protein, and that process is called translation. Okay, so there is a protein formation from the DNA through RNA, and that is called the central dogma. Is it clear about central dogma? And now next we can see, so here the DNA can transcribe mRNA or RNA strand, and then it translates into protein. So this is the central dogma. And one more thing I want to tell you. Here, if a DNA single strand is having the polarity 5', 3', and the nitrogen bases are A, C, G, A, when it undergoes replication, the next newly formed strand must have 3', 5' polarity with the nitrogen bases thymine, guanine, cytosine and thymine again. But when this strand form an mRNA, here there is a change in the nitrogen bases. The polarity must be anti-parallel. It must have 3', 5' polarity. But arginine base pair with the thymine. But in case of RNA, thymine is not there. Instead of thymine, I taught you which nitrogen base is there, which pyrimidine is there? Uracil. Okay. And here the same guanine, cytosine, again uracil. So it is RNA because of having uracil. Okay. So DNA contains thymine, but RNA contains uracil. When the nitrogen basis of one strand is known, it is very easy to predict the nitrogen basis of another strand in DNA. And in RNA also we can easily predict if we know the template strand nitrogen bases. Is it clear? Next we can see central dogma reverse. In some viruses there is a flow of genetic material from RNA to DNA. From RNA to DNA. Normally there is a flow of genetic information from DNA to RNA, but in some viruses, example retroviruses, there is a flow of genetic material from RNA to DNA, and that central dogma is called central dogma reverse. Central dogma reverse. And here the transcription occurs, and that transcription is called a reverse transcription. The reverse transcription. So it is a peculiarity of this retroviruses. Hope you understood everything about central dogma. That's all for today. Thank you.